So for chapter 16, we're going to focus on autoimmunity. So we want to remember that we've talked about tolerance throughout the course of the semester, especially when we're talking about T cells and B cells and their development. And so that plays into autoimmunity, because when there's a dysregulation of it, that's one of the theories of how autoimmunity arises. So with tolerance, remember, we want to prevent the immune response against self-structures, so self-molecules, self-proteins. And we have central tolerance, where we delete lymphocytes before they mature. So this is where we'd have negative selection. So this is going to happen in the lymph node organs where they're generated. Now, peripheral tolerance is once it goes out into circulation, once it's in the lymph nodes, the spleen, how can we stop any of those that might have gotten out? Or if there's been some kind of somatic hypermutation, so now the antibody might recognize self, how can we delete those so that won't recognize self and cause autoimmunity? Okay, so again, we've talked a lot about central tolerances. We've hit on peripheral tolerance in some ways. Again, as you can see in the figure, you can see how there's that Goldilocks of just binding just right, that if you bind too strong or too weak, you would go into apoptosis or become anergic. So one of the ways in which peripheral tolerance is regulated is with regulatory cells. And the regulatory cell that's focused on the most is regulatory CD4 T cells. So keep in mind, CD4 T cells are helper cells, and so within that population there's ones that are regulatory in that they produce cytokines that decrease the response. So IL-10 is one of the major cytokines that's looked at as an inhibitory cytokine that's produced by T regulatory cells. T regulatory cells do have markers that can be used to identify them, so we can see the different subpopulations of CD4 T cells. So like FOXP3 is a common one that is looked at. So here in this figure, you can see how we have this effector T cell. It's producing IL-2. The T regulatory cell can see that through its receptor, and that's going to allow for it to produce inhibitory cytokines, telling that effector cell, no, don't respond, quiet down, you're producing too much IL-2, you're overreacting to the situation. And they can, the T regulatory cells can interact with antigen presenting cells because they're going to have a T cell receptor, and they're also going to have co-stimulatory factors, so they can get signal 1, signal 2, and signal 3, but again, their effect is going to be different than an effector T cell, a T cell that's going to become activated and then respond in a positive, activated, let's go out and destroy that manner. So we want to keep in mind this is always occurring. So there's an establishment of tolerance, and we think of that more in central tolerance, but also in peripheral tolerance. But then there's also maintenance of it. And again, this is where T regulatory cells are really important because they can actually have a bystander suppression effect, because they're going to produce these cytokines, and in that environment, it can have an effect on those effector cells. There are also CD8 regulatory T cells. So I think we could argue that they're not as well studied. So identification of CD4 regulatory T cells happened first. So then scientists then identified a population of CD8 T cells that are regulatory, that again can suppress an immune response. And so exactly understanding their role is still under investigation, and that would be an area that you could look at, like hot research coming out, and see what's new in that area. It's believed that they can act by lysing APCs. So if you kill APCs, they can't present. They can also inhibit APC function, and they can also have regulation on effector cells that bind to the same antigen. So again, it's different than how the CD4 T regulatory cells suppress activity. There are also regulatory B cells. They do produce IL-10, and again, an area for additional studying and understanding their roles and how they suppress a response. And there's also myeloid-derived suppressor cells, and they can secrete IL-10 and a couple other factors, and again, they can negatively impact autoimmunity. So again, allow for peripheral tolerance. So again, keep in mind with these, one of the things that is being studied is the population, the percent of these cells found in quote unquote normal people versus individuals with autoimmunity. And this is actually how T regulatory cells were first identified, was that they were lacking in individuals with autoimmunity, and so that kind of spoke to their role. All right, so looking more specifically at autoimmunity, so autoimmunity at the basic level is a failure of tolerance. So again, we have a number of mechanisms in order to make sure that cells that recognize self are removed, and a failure at some point of those processes is where we can get autoimmunity. Now, autoimmunity includes a number of different diseases and disorders, and they can be organ specific or they could be systemic. So a lot of that depends upon what the antibodies recognize, if there's antibodies, what the T cells recognize, what immune complexes that are formed recognize. And so depending upon if it's targeted to a protein, where is that protein being expressed? Is that protein only expressed in a certain cell? Is it only expressed during a certain time in development or a different stage of an individual? And that, a lot of that can dictate when onset is, or what's known as triggering events. So these are a table from the textbook looking at some of the organ-specific autoimmune diseases. So I'm going to go to type 1 diabetes. So that targets the pancreatic beta cells that produce insulin. So T cells and also B cells producing autoantibodies recognize proteins that are expressed in the beta cells. And then that causes an immune response to kill the beta cells, which means that the individual doesn't have insulin producing cells, so they can't regulate their blood glucose levels. Again, I'm just going to pick that one out, but there's a number of different ones. There's also slides at the end of the presentation that go through the specific diseases that you can look at. There's also systemic autoimmune diseases. So these ones can have varied symptoms depending upon what the target is. So multiple sclerosis is one you might be familiar with. So this actually affects brain matter, and depending upon what part of the brain is affected can give you different symptoms. So this is Th1, cytotoxic T cells, and autoantibodies are found in these individuals. Also, like lupus has a number of different targets, so anything from nucleic acids to nuclear proteins. And again, we can see autoantibodies and also immune complexes that are formed. And again, there's a number of additional ones that are on here. So to study autoimmunity, there are several animal models that are used. So if you, earlier in the semester when we looked at the Jackson labs and their database of animals available, you might have stumbled upon an autoimmune model. If you didn't, you could go back to that website and you could look for different animals that are used for autoimmune diseases. So you could put a disease in instead, and then you could study or you could look at the information on those different mouse models. If you go into autoimmune research, you would definitely most likely end up working with mouse models, but there are other animal models that can be used. Some of them are spontaneous induction, so there's usually some kind of genetic factor that leads to this leakiness, this lack of tolerance. So the non-obese diabetic mouse, or NOD mouse, is commonly used for type 1 diabetes. So this mouse will spontaneously become diabetic. A higher percentage of female mice will become diabetic versus male mice. So sometimes there's a little discrepancy. It's not 100%, but by 6 weeks of age, about 80% of the mice do become diabetic. So you can do screening of the mice, and then you can also look at antibody responses and such. What's interesting with some of these models is that you can actually take T cells from one of the mice and you can then transfer them in, and the T cells alone can actually induce disease. So that's what I looked at was after we'd identified, other labs had identified T cells that could induce diabetes in these mice. I looked to find out what the antigen was, what was the T cell receptor binding to. There's also experimentally induced autoimmune diseases. So this is where you have a mouse and then you give them antigen. So either in an injection delivery or delivery in their water, you give them the inducing antigen, and then that induces the disease. Okay, so we want to remember that thinking back to how central tolerance arose, there are certain genes that need to be expressed. Also, if we look at the peripheral tolerance, there's certain genes that are going to be expressed, like in our regulatory cells. And so there are a couple different examples of known genetic factors that play a role in developing autoimmunity. So I just want to, disclaimer, for most individuals, so with any one of these autoimmune diseases, there's going to be different subsets. So it's not going to be a one answer fits everybody situation. And so if we look at type 1 diabetes, there may be some individuals that develop type 1 diabetes because of a genetic mutation to their AIR gene or a genetic mutation to FOXP3 gene. But that's not going to explain 100% of the individuals with type 1 diabetes. So again, just to kind of think of future direction, when we look at therapies, one therapy for everybody is not going to be probably the answer. More customized therapy, looking at genetic analysis and then coming up with therapy. So little jumping ahead of myself there, but again, just to kind of keep that in mind when I'm talking about this, it's not like, oh, this is what if you know someone with type 1 diabetes, this is what must cause it. Most individuals, it's going to be multifactorial. So there might be some kind of genetic predisposition to it. But again, there's going to be some kind of triggering event that's actually going to induce the diabetes. So we do know there's a couple classic examples where we see this disruption in the immune system's normal regulation. So that there is APS1 and there's also IPEX syndrome. So these are two classic examples where there's a disruption of the normal immune regulation. There's also some monogenetic disorders. So this is where a single gene is mutated and that single mutation is enough to cause autoimmunity. So that's the AIR gene and FOXP3. So again, keep in mind AIR is going to affect central tolerance. So remember AIR, that's what allows your thymus to produce and your bone marrow to produce all of those different proteins and process them and then present them during negative selection. And then FOXP3 gene is going to be your regulatory cells. If you have these, there's going to be multiple organs affected because this is going to be a systemic disruption of your immune system.